dendrites. Equal gray matter. So when we look at the spinal cord, then this gray matter here are cell bodies and dendrites primarily, and the white matter are nerve fibers. Okay, the long axon, or if we look at a pseudo unipolar neuron, which is sensory. Then we have a neuron that looks like this. It has a peripheral process and a central process. This coming, say, from the skin or your joint, and this going into the spinal cord. So these processes would be myelinated. But the cell bodies would not be. Okay, so the same thing. Cell bodies are gray matter, the axons or the peripheral processes are white matter. Now I'm going to do a coronal section of the brain and a cross section of the spinal cord and relate this to those structures as well. And we'll go over that some more on Thursday when we start talking about the cerebral hemispheres. Okay. So we have a cross section of the spinal cord here. So I'll diagram that. But before I do, I want to do a coronal section of the brain. So we have the foldings of our. We'll make this something very smart. We'll put it just in foldings. It's cow brain instead of human brain. So gray matter in the brain is found on the outside edge. This is going to be the ventricle. So we have lateral ventricle and third ventricle. And then within the white matter, we also have more gray matter. So those of you that might be started working on various brain diseases, we have the thalamus here, hypothalamus, and we have some other gray matter here. We have gray matter inside the brain as well. Okay. And then the rest of this would all be white matter. So if I were to draw this with our neuron, here would be a motor neuron. Use a different color. Here would be a motor neuron. This is all for free. We're going to get to explaining this part. I'm just trying to illustrate gray and white at this point. Cell body is going to come through here like this and go down the spinal cord. All right? So gray matter, because the cell body is in the cerebral cortex. That's what makes it gray. It's passing through this area, so all of this would be white. Okay? Coming up from the spinal cord, we would have sensory neurons. They're going to come up and stop in the thalamus. And then, now we're going to be 2 inches posterior to this, but I'm not going to be thinking about that. End up in the brain. So notice what the thalamus is made up of? Cell bodies. And so it also is gray matter. This is inaccurate. Our motor cell bodies are not in the same part of the brain as our sensory. I'm just kind of overlapping this to give you an illustration. Okay? So the gray matter in the brain is cortical. So this is cortical gray matter. And then we find it in these islets or islands, and these are known as nuclei. Gray matter, which are islands of cell bodies. Okay, gray matter. White matter. Myelinated nerve fibers. Okay. Whenever we talk about gray matter, we're talking about cell bodies. Whenever we talk about white matter, we're talking about myelinated nerve fibers, the axons or the processes. Now there might be some that aren't myelinated, but there are enough to appear myelinated. So that's the brain. When we look at those same white and gray matter regions in the spinal cord, again, I'm not going to label these parts of the spinal cord. Okay, so the H-shaped structure is gray, and all the stuff around it is white. So for instance, okay, this motor neuron that's coming down here is going to come into the spinal cord, and it's going to synapse with the cell body. That cell body is going to send its neuron out the spinal cord. That's why this is gray, because it's the cell bodies of these multipolar neurons. Okay. Now a sensory neuron is going to come in. Its cell body is out here, and it's going to come in and synapse with another cell body here. So we have gray matter here. Notice we also have this gray matter outside the central nervous system. So what do we call this gray matter that was inside the central nervous system? Nuclei. Okay? A collection of gray matter outside the central nervous system is known as a ganglion. Gray matter, cell bodies, outside the central nervous system. So in the morning lab, I have some students doing their case study on a comparison between Alzheimer's and Pick's disease, and another group doing it on Parkinson's. Parkinson's involves these cells right here. And they keep talking about the basal ganglia, which is an old term. The proper term is basal nuclei, because ganglia are cell bodies outside the central nervous system. Okay? So just some housekeeping here for terminology before we come back to structures of the spinal cord. So when I talk about just gray matter, the central peripheral processes. All right. So now let's go back to the spinal cord. Can I erase the board? Almost. Okay. So the spinal cord begins at the level of the foramen magnum. Inside the skull is the brainstem, and that's not arbitrary. We don't call it the same thing found inside the skull, the brainstem, and half an inch later when it's outside the skull, it's the spinal cord. If you look at a cross-section, it actually does look different. So the internal anatomy of that is different. So the spinal cord extends from the foramen magnum down to the level of L1, L2 vertebra. I'm going to draw it shorter and fatter than it actually is in proportion. It has a swelling at the cervical region, another swelling in the lumbar region. And it ends by coming to a point. All right, this swelling is known as the cervical enlargement. And this swelling is known as the lumbar enlargement. They're not, as I said, they're not really as dilated an area as I indicated on the diagram. But this model that I have up here, one of those really long front desks, is a great model of the spinal cord in place. Right here is the cervical enlargement, and here is the lumbar enlargement. Why do you think the spinal cord is slightly wider in diameter in those areas?
Lots of cell bodies.
Yeah, exactly right, and lots of nerve fibers. But why?
Because it's hesitant.
Yeah. So we have lots of cell bodies in the motor areas for going out to skeletal muscles, like I drew on the over there, and lots of white fibers coming in, bringing in sensory information. So the cervical and lumbar enlargements are for the upper and lower limb motor and sensory. The end of the spinal cord, where this comes to a point, is known as the conus medullaris. Oops, it should be 2 L's. And again, that ends at about L1, L2. Yes?
Did you say how many vertebrae there are wide or something, or is it just a thin vertebrae?
I'm not sure what you mean by vertebrae.
The cervical enlargement.
Oh, cervical enlargement is from like C5 to C8,
Okay.
and lumbar is T10 to L2.
Okay, so there.
Yeah. Now we have pairs of spinal nerves exiting between the vertebra, remember the intervertebral foramina, and there are 31 of these. We will talk about these in our last unit with muscles, but they're going to come out and pass through the openings of the intervertebral foramina, like you see here. Now when we get to L2, we still have L3 and L4 and S1, S2, S3, S4, S5 spinal nerves. But the spinal cord is already stopped. So those spinal nerves are going to continue down to exit from their appropriate vertebra. And they're going to continue down below the spinal ending of the spinal cord. And so this continuation of spinal nerves looks like a horse's tail. Remember the sacral canal contains a lot of these spinal nerves as well before they exit. And so this is known as the cauda, which means tail, equina, which means horse. Those are all nerves, all right? So these are spinal nerves. Now I'm going to leave that diagram up and draw just the spinal cord so we can look at the meninges without these extra structures on. We can add the layers of the covering to the brain and spinal cord. So the next page in your lecture notes is what we're going to not cover right now, and you're not responsible yet on a lab quiz for that information, the components of a cross-section appearance of the spinal cord. That's supposed to be today's lecture. So instead, I want to move ahead to the meninges. So we've done page 32. So turn to page 34, and what I'm ignoring is page 36. Okay. So I'm going to draw the brain over here. Brain stem. A very fat and a very short spinal cord. Now let me look ahead. I did not use the same color scheme this morning. I think it'll be easier if I do. So you'll notice that each of the layers of the meninges is called a mater, and mater means mother. So the closest, the deepest layer, the one directly in contact with the spinal cord, is known as the pia mater. So this is going to cover intimately the surfaces of the brain. It's going to go into all the folds. It's going to come down the spinal cord. It's even going to get folded into the brain and into the ventricular area. We'll come back and talk about that in a moment. Along the spinal cord, it gets pulled out into little tent-like structures, little points, and attaches to the other layers of the meninges. And these kind of help to form the support, stabilize the spinal cord so it doesn't shift from left to right within the spinal cord. And then at the very bottom, a single layer pia mater extends all the way down to the coccyx. So we have our coccyx level here. I just am writing coccyx at the bottom. This is L1 and L2. These structures are known as denticulate ligaments. And there is a misprint on page 32 of your lecture, 34 of your lecture notes. So on a pia mater, it says denticulate ligament, and then it repeats pia mater. That second pia mater should say filum terminale. And that is what this piece of pia mater goes terminale, and extends from the spinal cord, the conus medullaris, through all those spinal nerves down to the coccyx. Okay, so it's not a nerve fiber, it's found within the cauda equina, but it's not a nerve fiber. It's just a piece of that pia mater connective tissue. So let me underline in green these two terms to reinforce that they are made up of pia mater. The first time I drew them in red because they contain the blood vessels on the surface of the spinal cord and brain. But the other diagram that's in here shows it in green, so I can try to stay with that. So the pia mater is this most intimate covering that contains the surface blood vessels right here. And you've probably heard of a subdural space, a subdural hematoma maybe, where there's bleeding onto the surface of the brain. And it's actually a subarachnoid space. Because our middle layer is the arachnoid layer. What does arachnoid refer to? Spiders. And so what actually happens is there are collagenous fibers extending across this space to the pia mater. So I'm going to exaggerate that gap. I'm going to draw this in. And this goes down to S2. And so we have these fine spidery lines that extend from the deep surface of the arachnoid matter. And either someone thought they look like spider legs or they look like cobwebs, probably the latter. And this also goes all the way around the brain. It doesn't go into every little fold. So this would be the arachnoid matter, or the spider mother, what we always wanted. And the space between the pia mater and the arachnoid matter is the subarachnoid space. And it contains CSF. So later in lecture we'll talk about how the CSF fits there. CSF is cerebral spinal fluid. So that's going to be between the pia mater and the arachnoid matter. So if I were to illustrate it on here, this would be pia mater. Here would be arachnoid matter. And this would be the subarachnoid space. So again, that's around the spinal cord, that's around the brain. And so the subarachnoid space is allows a continuous flow of CSF. The outermost layer is called dura mater, or the tough matter. So remember when we talked about synovial joints, we had the synovial membrane, which is very delicate, and then around that we had the synovial capsule, fibrous capsule. So this is like adding a fibrous capsule to the dura mater. And in fact, the arachnoid matter and dura mater are attached to each other. So dura mater and arachnoid matter both end in S2. So this would be the pia mater, arachnoid matter, and dura mater. The subarachnoid space, this one, is between these two. See, the diagram you're looking at is this one here. So it's pulled back. The arachnoid is on the surface. If I were to peel it back, the arachnoid would be on the deep surface of the dura mater here. And then the pia mater is attached to the spinal cord itself. Okay? So here, let me turn the lights off a little bit. This is a diagram from Netter's. Netter's atlas is really good for the nervous system. So if you don't have one yet, you can get a paperback copy or flashcards for them, or you can find some of these images online. So the dura mater has been cut open. Here's the arachnoid matter, and then down the lower end of the screen, you can see the, let me move this up so those of you that are, the lines won't move up, but I can move this up at least. So here's the denticulate ligament right here, and right here, where pia mater is pulled out and attached to arachnoid and dura mater. So here is a real cadaver where the vertebrae and spinous process has been removed, dura mater's been cut. Here's arachnoid matter, and then you can see a little bit of denticulate ligaments here. On one of our cadavers, we have that exposed, so I'll see if I can bring Thomas out for lab next week. And we have Thomas's brain also. Now this is the model that I showed you up front. Okay, here is the top with the pons. Here would be the cervical enlargement, and here are the spinal nerves passing through the intervertebral foramina. And it's hard to see, but on this one side up here, we have the denticulate ligament. If we look at the lower end, here's the conus medullaris. Here, the phylum terminale has been painted, but on our model, it's not. I don't know if it's been painted or somebody just photoshopped it in here. But if you go to the very tip of the conus medullaris, then you can follow that down as the pia mater. Whereas all these other structures here are nerve fibers that form the caudate equinum. Here's this white line right here is the dura mater and arachnoid matter, stopping at S2, while the phylum terminale continues down to the coccyx. Okay? We're not doing the cross section yet, so we're going to move ahead. This just shows the spinal cord in situ, meaning in place. So this is a cadaver from which the frontal material has been removed. Here's the body of a vertebra. Here's the spinal cord with the motor and sensory neurons coming together and then passing to supply the deep muscles and skin of the back. So this would be the intervertebral foramina that those spinal nerves are passing through. And then this is just a model showing the same thing in the same position. So body of the vertebra, spinal cord, spinal nerve coming in and out. You can see the gray matter and white matter. All right, so now let's look at these coverings if they extend around the brain. So the same tissues, we're going to be looking at the area up here of the brain. All right, so this area right here on the brain, top of the brain, is the same as the very top of my diagram. And so here's this is where I got the, this is not quite the same colors as I used, but here's the pia mater in blue. Notice the blood vessels on the surface of the pia mater. Here's the arachnoid matter in purple, showing the subarachnoid space. And then above that in brown is the dura mater. Okay. So now we're going to focus on the dura mater as it covers the brain. Where it covers the brain, as I showed right here, it comes down between the two hemispheres And here are the two hemispheres. So a hemisphere is half of a brain, right? Cerebral hemisphere, telencephalon, and the diencephalon is going to be here in the center. So when we have this half of a brain, as we'll talk about on Thursday, there are fibers that connect one side of the brain to the other. So frontal lobe to frontal lobe, temporal lobe to temporal lobe, parietal lobe to parietal lobe. And they form this C-shaped band of white matter that we'll talk about on Thursday. The point I'm trying to make is that there is a gap between those two halves of the brain that we can actually, on our sheet brains, we'll see as well. And this is the dura mater, okay? The dura mater, well, it's not really, but you know what I mean. The dura mater comes down between each half of the brain like this as a fold, and then covers the brain around that way. So this is what we call a dural fold, all right? The same thing occurs between the brain and the cerebellum. So between the brain and the cerebellum is a fold of dura mater. And then there's a really shallow fold between the two halves of the cerebellum. So that's known as a dural fold, and they form this shape right here. So where it forms, remember I said that the white matter going back and forth from left to right was a C shape? So we see that this fold forms a C shape as it goes over the connecting white matter. Here's the fold where it's found between the occipital lobe of the brain, and underneath would be where the cerebellum fits. So what I'm going to diagram on the board for these folds looks like this. All right? So on each half of the brain, that would be where the falx cerebri, or this middle fold, fits down inside the gap between the two hemispheres. It has this C shape. All right. Here is the tentorium cerebelli, and again, I will label them. And the cerebellum. Actually, I should say fits on either side. The bottom fits on either side like this, and the occipital lobe will be on the top part. Okay, so that creates another structural support that keeps the brain from shifting too much. And in fact, the front of this right here is attached to the crista galli. Right at this point right here, that's going to fit over and attach to the crista galli that we talked about with the ethmoid bone. So may I erase this half of the board? You guys okay with that? So as we look at this structure from anterior to posterior, has this C shape. And this is known as the falx, which does not mean false. It means fold. Okay? So this is the falx cerebri, or the cerebral fold between the two cerebral hemispheres. And then there is a kind of a tent shape. And this is known as the tentorium cerebelli because of that tent appearance. So on the model, this would be the falx cerebri. And this right here would be the tentorium cerebelli, with the cerebellum below and the occipital lobe of the brain above. there is a small fold. It's very, very, very shallow, mostly in the posterior region, which slightly separates the cerebellum. So, kind of like this. Inferior, which is the false cerebellum. So we have two cerebellis and one cerebri. So these are, again, folds of dura mater that separate these components of the brain. You can see them. I'm going to turn the lights off here just to... So here in the diagram, here in the, this would be false cerebri, tentorium cerebelli. And the false cerebelli is really hard to see again. False cerebri, tentorium cerebelli here. I think that's the last name. So I used to, we were trying to figure this out at home before we had these models. I would make it out of paper to try to illustrate to students what this actual shape was like. So that would be the false cerebri, and then the false cerebelli, tentorium cerebelli. How that?
This is just basically a stabilizer for the brain.
So up here would be all the lobes, frontal, middle, parietal, occipital lobes. Down here would be the cerebellum. And then this little piece right here would be the false cerebelli. But because this is paper, but it's just very shallow between the two halves of the cerebellum. Okay? Now we have a real one right here. So this is part, it's a very small brain. So this is part of the dura mater. Attached to the inside would be arachnoid matter, but we don't have the spidery leg web part of it anymore. But then this would be part of the false cerebelli right here. And again, it would fit over the brain in this fashion, where the two cerebral hemispheres are on either side of that false cerebelli. Okay? False cerebri, which is in between the two. All right, so now let's look at, these are the meningeal structures. And you'll notice in this view right here, this would be the false cerebri right here. Okay, so it's this structure cut in cross section, same as right here. So that is that stabilizing dura mater that's found between the two hemispheres. And you'll notice that at the top and the bottom, it's split. So it's really not a vein. It's really not a blood vessel. It's a split in the dura mater. Okay, so in that fold, where it came together, there is this triangular split up here and down here. And these are two of what we identify as dural venous sinuses. They're not blood vessels, but they receive CSF. So the CSF is constantly being made, and they receive blood from the brain. And they send both of them, which came from the arteries, they send both of them back to the venous supply to go back to the heart to be recirculated through the body. So blood is constantly being delivered to the brain and supplying the tissues, and it has to go back to the heart to get more oxygen, into the digestive system to get more nutrients, and goes back to the brain again. The CSF, cerebrospinal fluid, is constantly being produced in the ventricles and leaving the ventricles and going into the CSF space, and so it has to go from there back to the blood as well. So we have blood and matter. We're going to look at these dural venous sinuses, and then we'll look at the blood supply and drainage of those two fluids. So I'm going to leave this diagram up on the board now and erase this one. And I'm going to draw this region right here larger. Okay, we're going to erase this diagram. You guys okay with that?
Has been recording.
This? No, it's broken. It just says preparing for like ever.
So this is dural matter going around the side of the brain. This is the fault cerebri. Okay, and now if we add our other meninges, pardon? Yes, the tip of it here, not this. This is that, okay, or that space. All right, so now I'm going to add the arachnoid matter. The arachnoid matter is attached to the inside of the dura mater. When it gets to this sinus, there's actually openings in the sinus, and the dura mater forms these kind of blossoming cauliflower-like structures. And these are known as arachnoid villi. You can see them a little bit in this picture right here.
Yeah. So, Venus look
So see them right here? This is what I'm drawing. So these are the arachnoid villi penetrating into the sinuses. And then we have our spidery lines in our subarachnoid space. And then we have our pia mater, which was green, right? These are the folds on the surface of the brain. So subarachnoid space. Remember CSF. Arachnoid matter. Pia mater. So here as we look on the surface, the dura mater is opened up, and you can see that on the surface of the brain, we have these blood vessels, again, associated with the pia mater. So blood, and we'll talk about how blood gets into the brain. We'll go back to the carotid arteries and vertebral arteries we talked about with the skull. So those arteries go into the brain, form capillaries, give off their nourishments, give off their oxygen, come back to the surface of the brain, and form these cerebral veins. And notice where they're going. They're going going into this venous sinus, which is really not a blood vessel, so we're not calling it a vein, but it's this split in the dura mater. So in addition to having the CSF and the T matter, we're going to have blood vessels coming out of the brain and then traveling along the surface and emptying into the venous sinus. So it's delivering, these are cerebral veins, and they're delivering deoxygenated blood that's been used by the brain. So you can see this channel that I've drawn right here, running here along the midline of the dura mater, just over the top of the space between the two cerebral hemispheres. And so this is identified, we're going to see it here in our falx cerebri. That's the top one. This one here is going to be the bottom one. And those are identified as the superior and inferior sagittal sinuses. They're not veins because they're in the dura mater. There's spaces in the dura mater. So this is the superior, it's on top. Sagittal, it's in the midline sinus. Okay. And this one on the bottom is the inferior sagittal sinus. That would be this one here. And this one would be the superior. So on our model, see this right here? Superior sagittal sinus. These little circles are the arachnoid villi. Here would be a smaller inferior sagittal sinus. Okay? They are a split in the dura mater. So the dura mater separates. They're not lined by any epithelium. It's just an opening, a split in the dura mater.
But I mean, is there just hollow, or do they have,
The only they're hollow. Yes, they have fluid in them. I'm sorry. They have the CSF and they have the draining venous sinus. So here's what I just drew on the board. Let me make that a little higher.
Depends what's this one in the dark or something more smoke. It was dark.
All right. So here's what I drew. This is an enlargement of what I drew here. So here would be cerebral veins coming up out of the brain tissue, passing through the pia mater, the arachnoid mater, and opening into this split of dura mater. And this is the superior sagittal sinus. Here would be arachnoid villi.
and there's dura mater on
Above and below.
The whole thing.
Yeah. So here's the dura mater. This is skull.
Yeah.
Here's the dura mater splitting
Okay.
to create the sagittal sinus. Here's the falx cerebri, and then coming back together again. All right.
And the
On the other side.
cerebri is just 2 layers with the dura
Uh-huh. Yeah.
fused
Yeah. So they come together, and that would be that split, and then they fuse on either side. Okay.
So if we have internal hemorrhaging and they want to do like a tap for, they're going for one of those.
They're looking for subarachnoid, now it depends, it could be internal hemorrhaging within the brain itself.
Right.
Okay, but usually when they talk about a subdural hematoma,
Mm-hmm.
so is arachnoid. So that's a subdural hematoma.
Now would that flow into one of the sinuses though? Or?
No, it's what it's going to, it could, some of it could.
Okay.
Okay, most of what happens is when we get a bruise, which is what a hematoma is, and the tissue, the blood vessels are broken, we get a big black and blue of swelling. So my son had his last football game Sunday morning, and he's a center, and he's always wide open because the quarterbacks, you know, have their favorite wide receivers. And so he's, I mean, 9 times out of 10, he's standing there, nobody around him for 40 feet, and he refuses to yell. We keep telling him, yell, at least that'll pull defenders off of the wide receiver. And, you know, he's wide open. Well, the very last play of the last game Sunday morning, this is flag football, so there's no tackle here. He gets the ball thrown to him. He pulls it out with one hand. At the same time that he's pulling it in, a defender comes up and slams into him with his elbow right here, knocks all the air out, and he hits the ground on his hip. I mean, just solid right on his hip. Okay, so he's got a nice, great, big, what do they call it, hip pointers right there. He did that, by the way, he kept calling. And that's going to form a big bruise because as the blood seeps into the connective tissue, that tissue is going to swell out. Well, in the brain, the skull doesn't swell out. So that's what furthers a concussion, if you will, and it's going to push the brain tissue in, and then you get compromised brain function. So loss of memory is part of that. It's not just, so with swelling, which is a simple concussion, there is bruising. So you're tearing these vessels here, as well as along in the brain itself, and that is increasing swelling. But if you have a lot of bleeding, because these larger blood vessels are torn, there's no more place for that blood
Right,
to
it's
go.
the pressure.
Yeah.
They-
So that's why
Sight, smell.
someone who has brain trauma, you don't want to give them aspirin because
Thins the blood.
that's just going to increase the flow of the blood. Okay, so superior sagittal sinus, inferior sagittal sinus. And then, of course, obviously, we need to get this blood back to the brain. And I think we'll take a break before we, yeah. So, let's take a break. And so, may I erase the board? You guys have this stuff by now? Okay. I'm going to come back and redraw the diagram on the left. So, I can send it to you. Did I erase too soon?
Nope. Thank you for always asking. I really appreciate it before you start swiping.
All right, so we have 2 sets of blood vessels. We talked about the skull that take blood to the brain. If you recall, the largest of the pair of blood vessels are the internal carotid arteries. Okay? And then the smaller pair are the vertebral arteries. Hold on, just 1 second. Let me see if I can. I meant to borrow this model while I was next door. Oh yeah.
And then just the bereaved part.
And landscapes made a noise. You had to get the whole sentence.
Oh, okay.
Well, it sounds to me like you have selective hearing.
I just might.
So we have this head model over on the side of the room, but it doesn't show the neck vessel. And I tried to find this, but there was lecture going on when we were doing the skull tissue. So this vessel right here is the common carotid artery. It is this one right here. Okay. Comes off major branches of the aorta as they come out of the heart. And the common carotid artery, and this you don't have to know this particular level of just the internal carotid. I'm just kind of showing you where it's coming from. Right about the angle of the jaw, this divides. And it forms an external carotid artery that comes onto the surface of your face and supplies, your mouth and the muscles that you chew with and everything. That's the external carotid. And then underneath, it's going to come up and come through the skull as the internal carotid artery through the carotid canal. So we talked about that in lecture with the skull. And so right here, this vessel right here is the internal carotid artery coming through the carotid canal. Okay? And so that's the major supply of blood. Let me show it to the camera here. Okay, right there. That's the major supply of blood to the brain tissue itself. So a pair of internal carotid arteries. Coming through the carotid canal, kind of making a bend and entering the skull there. Well, they're surrounded by the musculature of the neck, so they are deep. The common carotid is not. You can kind of feel that under the sternocleidomastoid mass and the others are even deeper. Okay? So that's the major blood supply to most of the brain. It's from the source of the internal carotid artery. And part of the reason it helps to see this in here is when we look at these, we look at an inferior view of the skull. And sometimes getting that 3-dimension is a little bit difficult. So if you're having trouble during lecture seeing that, then take a look at one of these skulls. I will have both of them here. Okay? So that's internal carotid artery, a pair of those through the carotid canal. And then you'll notice here, running up along the cervical vertebra, we have the vertebral arteries. The vertebral arteries come in through the foramen magnum. They're much smaller, because if you remember looking at our cervical vertebra, they don't have very large diameter. So those are the only two vessels that supply blood to the brain. The middle meningeal artery, coming through the foramen spinosum, is supplying blood to the meninges, into the skull. It's not supplying blood to the brain. So we have the internal carotid here. That's showing middle cerebral, but coming into the brain, the internal carotid and the vertebrals. We're going to look at how those branch to further supply the brain tissue. So this is now looking at the inferior surface. So looking at the brain turned upside down. The neck would be here. Here is the pons and the medulla of the brainstem. To reiterate, this would be from the myelin and metin and myelencephalon of the rhombencephalon, the pons and medulla. And here is the midbrain, barely visible, of the mesencephalon, and this is diencephalon area here. So coming through the foramen magnum, along the spinal cord, we have the two vertebral arteries, and I'm going to diagram and label this on the board. They join together to form the basilar artery. This is called the basilar artery because it crosses the basal pons. When we get to basal pons next week, it's what I call the pregnant belly part of the pons, and not to leave you guys out, you can also call it the beer belly part. Okay? And then it will end by dividing to form these two major vessels right here. So that's all vertebral artery. And then here is the cut surface of the internal carotids. So remember, they're coming in like this. So this is after this has been cut inside the skull. And so we have this cut surface of the internal carotid. So you can see this, it's kind of reversed, but you can see this on this model by looking inside. Instead of looking on the surface of the brain, you're looking on the floor of the cranial cavity, and the pattern will be the same. So again, on this view, you can see the internal carotid artery Remember the external going to face. There's internal coming up and doing most of the brain, while the vertebral arteries come up and do cerebellum and the back of the brain. All right. So let's diagram that, label it, and I'll also include what parts of the brain are being received. The old name for this is circle of Willis. We currently call it the cerebral circuit. And while I have this diagram up here before I start diagramming it, let's say that you have atherosclerosis. You have a lot of fatty plaques building up in the smooth muscle of your arterial wall, and so your right carotid artery is completely blocked off. That doesn't mean that the right side of your brain doesn't get, it would be this side, doesn't get any blood supply, because we have coming up the vertebral arteries. Blood can communicate through here and go into the middle cerebral artery, even though this is completely blocked off. All right, or it can come in this one and go around and then get to the brain. So most of our joints, our shoulder joint, our hip joint, our elbow, our knee joints, have lots of blood vessels branching around them like this. So if one gets blocked, it could go around and still continue on. Interesting enough, the heart doesn't have this kind of going around ability. Blood vessels from the front of the heart and blood vessels from the back of the heart don't meet. So if you have a blockage, it can't get blood to that part of the heart from some other vessel. You would think the heart would be an important enough organ that we would see that. Yes?
What are some of the advantages that enjoys?
For the heart?
Yeah.
I don't know. I mean, when you exercise, you develop more blood vessels and they get closer to each other, but they never actually connect. And so you get more and more and finer blood vessels, and one of the ways of treating for heart attacks is they actually take a machine with a laser and they burn holes into the muscle wall of the heart to create more blood vessel stimulation, of more blood vessel growth. But they don't actually, and I don't know what the advantage of that is. It just seems to be a huge disadvantage. Let's draw this on the board. And I'm going to start with the vertebral artery. So the vertebral arteries come up through the foramen magnum. And on the surface of the brainstem, on the medulla, they're going to join together. And form a common vessel. Before they join, a major branch, there's lots of branches given off here. But the vertebral arteries are going to be traveling along the medulla. And their branches will supply the medulla as well. My abbreviation for artery, by the way, is an A. And my abbreviation for arteries, plural, is 2 A's. So you see that. This vessel is known as the posterior inferior cerebellar artery. Branches that I haven't drawn in for the vertebral arteries are going to supply the medulla. Posterior inferior cerebellar artery supplies the medulla and the cerebellum, as you can see in its name, which is abbreviated pica. And this is not the Rocky Mountain rodent. These are particularly important because, as we'll see next week, the medulla is where we have the cardiovascular and respiratory centers. If you have an aneurysm here or a blockage of these blood vessels, it's pretty much instant death, all right, because it stops the reflexive stimuli to your lungs and stops the heart, essentially. Then these two join together and create the basilar artery. And branches from this will supply the pons and the cerebellum. The pons again is part of our brainstem. And then these end by forming a T. Known as the posterior cerebral. So we're going to have 3 cerebral arteries, an anterior, a middle, and a posterior. There are 2 posterior cerebral arteries. And this goes to the back of the brain, the occipital lobe, and to the cerebellum. So that's all from vertebral arteries. Okay? So here are 2 vertebral arteries, and the basilar artery, and then wrapping around between midbrain and pons as the posterior cerebral. So that's what we'll see right here. We have a diagram of this to label in the lab study guide part. There are several arteries in there that you don't need to know, such as the ophthalmic artery and so on, just because they were part of the diagram. And I usually put an asterisk by them. I didn't. But the ones I'm drawing on the board and labeling are the only ones you need to know. Okay, so there's 3 or 4 that are on that diagram that you don't need to know. So these are the branches and blood and regions of the brain supplied by the vertebral arteries. Then we have the internal carotid arteries. And again, these are cut. They've come up through the carotid canal. And so this is upside down. So this is cut where it's come in through the carotid canal. All right? Has 2 major branches, and well, each one has a major branch and a very small branch. So I'm just going to put IC for internal carotid. Largest branch supplies most of the blood to the brain. If someone has a stroke or CVA, it's usually a problem with the middle cerebral artery or one of its branches. And I'll show it to you on the model here. So this is the middle cerebral artery. Right here, this would be this part of the brain. And it's going to pass between the temporal lobe and frontal lobe. So it's going to come out, it's going to come out here like this, pass around all of that, and it's going to pass through here, this groove right here, and it's going to do all of this part of the brain that my hand is holding on to. So I'll write it on the board: frontal lobe, parietal lobe, and temporal lobe. Remember, occipital lobe is done by the posterior cerebral artery. Okay? So up to about an inch or so from the middle part, all of that is done by middle cerebral. So if this gets blocked, you end up with paralysis. You will have speech problems. Okay? You may have personality changes. It all depends on where exactly in the middle cerebral artery that particular blockage is located. So frontal lobe, parietal, and temporal lobes, as well as thalamus and hypothalamus. It's easier to remember what's covered by the anterior cerebral and posterior cerebral, and everything else is middle, rather than label everything. Okay, now Now the two anterior cerebral arteries come together and meet in the midline. So right here are the two anterior cerebral arteries. These are the ophthalmic ones here, and they pass through between the two halves of the hemisphere where we have the false cerebri. And you can see them right here. So this is that band of white matter that connects the left and the right brains together. So the anterior cerebral artery goes over it and around towards the back and supplies this medial surface, kind of like this. Okay. So all of this medial surface and just a little bit over the edges is anterior cerebral artery. Essentially frontal and parietal lobes. Now notice that I don't have a connection yet. I have the blood flow of the anterior cerebral and middle cerebral from the internal carotid artery, and posterior cerebral from the vertebral arteries, but they're not yet connected. And so we have three communicating arteries that now make this a circuit. Between the internal carotid and posterior cerebral, we have two posterior communicating. And I'm not going to identify a part of the brain that's supplied because these are really more just connecting the other vessels. Okay? So this is also a posterior communicating. And then we have a single anterior communicating. Now we will do this again in Bio 431, and you already have it down. So two major blood vessels supplying, pairs of blood vessels supplying the brain, the vertebral arteries, but more importantly the internal carotid. They each form, so from those two pairs we get three pairs, anterior, middle, and posterior cerebral arteries, with the basilar and vertebral supplying the brainstem. Okay, now we have these blood vessels. Remember I said we were going to follow and see where the CSF fluid came from? So now we have these blood vessels, middle cerebral, anterior cerebral, posterior cerebral, along the surface of the brain, and they would be in the pia mater. So what you're seeing here are the blood vessels that are traveling within the pia mater. They're going to send smaller branches deep into the brain tissue to get to the white matter and the gray matter of the brain. Those will become smaller blood vessels, arterioles, and capillaries finally. Remember, capillaries have only a simple squamous epithelium, and those capillaries will then allow the oxygen and nutrients to move to the neurons and the neuroglial cells, and waste matter, carbon dioxide, and so on to be picked up and taken back into the veins. And the veins then will return to the surface, similar to the picture we ended before break, where in the pia mater we had our cerebral veins on the surface. All right, so that image that we looked at was this one. So we're going to come back and follow how this venous blood gets back into the sinuses. But some of these capillaries are going to become associated with the ependymal cells lining the ventricles and function to make cerebral spinal fluid. So if there aren't any questions on this, we're going to move back to formation of CSF on page 44 and 46. May I raise the board? You guys have that? Not yet? Yes. Yes, except this one isn't showing pica. Pica actually comes off the vertebral artery. And it comes out. It's not, you'd have to draw it in. It's in the diagram in your lab. Okay, everybody set? So this diagram that's on the board is the sagittal section of the brain and spinal cord. Back to those ventricles that we talked about on Thursday and reviewed this morning a little bit. And so, kind of shown in shadow here are the lateral ventricles. They will pass through the interventricular foramen into the third ventricle, which takes the shape of the thalamus, and the hypothalamus, which form its lateral borders. Pass through the midbrain as the cerebral aqueduct, and into the fourth ventricle. Okay? In the floor of the lateral ventricle, in the roof of the third ventricle, and near the openings of the fourth ventricle, we have this combination of capillaries from the pia mater and the ependymal cells. So that's what's being shown in the lower, the Christmas tree colors here. So the red is a capillary of pia mater, and the green are the ependymal cells, which if you may not recall from Thursday, but all of the fluid-filled spaces of the spinal cord and the brain are lined with ependymal cells. So the central canal and all of the ventricles are lined with ependymal cells. But only in those locations I indicated do we have the choroid plexus, where we have this special association with the capillaries and where we actually make CSF. So blood fluid, tissue fluid, not the red blood cells, but tissue fluid, leaves the capillaries, and as it passes through the ependymal cells, its content is changed. So the CSF has less glucose, it has less protein, and modified amounts of calcium and chloride and magnesium and so on. And so it's derived from the plasma, but it's not the same as. White blood cells can also pass out of the capillaries. Remember the term diapedesis, where they change shape and kind of squeeze out, and between the ependymal cells. So you can have white blood cells in the CSF. They increase in number if you have meningitis or a CSF infection. So the next slide I just found this morning, and this one kind of shows, not very clearly, but how the brain is developing, and as the developing lobes of the brain fold under, they incorporate the pia mater, and that's how we get the pia mater into the ventricles. Okay, and so this is where we see in the floor of the lateral and the roof of the third and the fourth ventricle, we have this choroid plexus. And again, the term choroid plexus, and I'll write it on the board in just a moment, refers to this combination of ependymal cells. So what you're seeing here in lavender purple are the ependymal cells, and here are blood cells of the blood vessels, okay, of the capillaries. So, choroid plexus is formed of ependymal cells and pia mater capillaries. Specifically found in the floor of the lateral ventricles, in the roof, the third ventricle, and the openings aperture means opening, so 2 lateral apertures and 1 median aperture. These go into the subarachnoid space. And then into the central canal. So on the next page is that same diagram I was showing you, so let's follow. Here's just a kind of a modeling with the computerized coloring of the choroid plexus. When we see it in the sheep brain, it looks like brown algae or seaweed. All right, so you'll actually be able to see it. So here's another illustration. We'll go back and forth between these two. Trying to get this like bigger. Oh well. So I'm going to turn the lights off and follow it through with you. Please move it up higher. So I'm going to start, I'm going to trace this. I'm going to start with the lateral ventricles, because obviously if we have CSF being formed in the third ventricle, it's not going to travel quite the same way. But here's the choroid plexus in the lateral ventricle. So CSF that's formed by that is going to travel through the interventricular foramen into the third ventricle. It's going to pick up more CSF from this choroid plexus and travel through the cerebral aqueduct into the fourth ventricle. Now remember, the fourth ventricle was diamond shaped. So on the sides of the diamond we have the 2 lateral apertures. And then just under the cerebellum here we have 1 single median aperture. So as it leaves that, the white arrows now are showing it in the subarachnoid space. It can also go down the central canal to the bottom of the spinal cord and come up the subarachnoid space. It's constantly being produced, about 150 mils a day. And we have to then have it leave the brain, because if it doesn't, you would get pressure on the brain just as if we had a bleed. So as it's in the subarachnoid space between the pia mater and arachnoid mater, it comes up here to the superior sagittal sinus and passes through these arachnoid villi into the superior sagittal sinus. Okay? Now, we also have our venous blood. Remember in the diagram I drew how the veins were on the surface? So those veins are emptying venous blood into the superior sagittal sinus. So now our sinuses have 2 fluids, both of which were derived from the blood, from the arteries. Venous blood and CSF. And they have to be returned to the cardiac cycle so that that volume is maintained constantly, and we replenish, we replace it with more CSF. So let's go back to those sinuses, and we'll see what happens at that, and then I'll diagram it to the trace. So here are the veins emptying into the superior sagittal sinus, as I showed you earlier. So this is how the 2 fluids get into the sinus. Here are cerebral veins carrying blood from the brain tissue to the sinus, and here are the arachnoid villi allowing CSF to get back to the sinuses. Okay. And then here we have the drainage. I don't know which is the clearer picture. We have this painted on the skull. So let me leave it with this one, and I'm going to diagram it. And then I'm going to actually verbally write out the sequence of structures that they go through. Right? So can I erase what's on the top of the board here? I'll repeat it. I'll pull the screen up and repeat it. So both our blood and CSF are entering into the superior sagittal sinus. So at the top, superior sagittal sinus. And we would have our arachnoid villi kind of draining into that. Then down here we have our inferior sagittal sinus, which is just, it's much more irregular and not as defined a space. And these are listed on the bottom of page 40. So now blood, or mixed blood and CSF flowing from the inferior sagittal sinus is going to flow along this baseline here. This one right here. Okay, so here's superior sagittal sinus. Inferior sagittal sinus is going to flow along this one, and that's called the straight sinus. And where they meet is called the confluence. Now, where are they going to originally, what is the first vein they actually flow into? Because remember, these are sinuses, not veins. So where is our direction? Where are we trying to get these? Internal jugular vein. Remember the jugular foramen? So that's where we're trying to get these sinuses. So when they get to the back, here at the confluence, they're going to either go flow to the right or flow to the left through the tentorium cerebelli, and those are known as the transverse sinuses. So each of these was only a single one, but now we have a pair of transverse sinuses. There's a transverse sinus, and there's a transverse sinus. And then they flow down the side of the skull, across a double curve. What's the Greek word letter for S? Anybody remember? Nobody joined a sorority yet? Sigma. So our curve is a sigmoid curve. So in the dura mater along the skull, we have a pair of sigmoid sinuses. And they end at the foramen, the jugular foramen. So finally then we have our internal jugular veins. Now there are other sinuses, petrous sinuses and cavernous sinuses, which you don't have to worry about, okay, that are more anterior. We have a couple of teaching skulls. These vessels create impressions in the osteoid of a forming intramembranous bone of the skull. And just remember the grooves of the middle meningeal artery we can see on the side of the temporal bone. So these have been painted to make them more clear, but you can see here would be where the superior sagittal sinus is found. Right? And then here on the back, the groove for the confluence, and then each transverse sinus. And if you look on this side, you can see the groove, which is actually about the size of your little finger or forefinger, for the sigmoid sinus, leading right to the jugular foramen. So you can see that is the transverse sinus. Now, why is there no groove for an inferior sagittal sinus? Brings back to the. Oh, it's on there. Okay, it's not affected with the skull because it's in the midline of the brain. All right. So these other ones we have those grooves for. And now, the cerebrospinal fluid and venous blood has been returned back to the cardiovascular system. The internal jugular vein will take it back to the superior vena cava and back to the heart where it can be recirculated again. So I'm going to pull the screen up and then just write this sequence on the board for you, okay? I'm going to leave that one up. I'm just going to pull up the other side of the screen. Let me just turn mine. So the sinuses contain venous blood and CSF, all right? So let's follow the blood source first. So no more new material today. We're just going to summarize this up, and then we'll have lap time. Give you back your exam. The blood source is two blood vessels, internal carotid arteries and vertebral arteries. Okay. And I'm not going to do the minor ones, but together these create the anterior cerebral artery, middle cerebral artery, and posterior cerebral artery. So all of that we've already covered. And then these will spread across the surface of the brain and send smaller blood vessels, smaller arteries, which will get even smaller and form arterioles, which will get even smaller and form capillaries. All of this we get next semester when we do cardiovascular systems, so you don't have to worry about this sequence. All right? The capillaries will then come together and form venules and veins, which will return to the surface of the brain and form our cerebral veins. And then those will empty into the dural sinuses. Okay? And you don't have to know any named cerebral veins. Those are just those blue vessels that I showed you on the surface of the brain emptying into the sinuses. Okay, then we had our CSF. That's the other fluid source of what we find in the dural venous sinuses. So for our CSF, we're going to start out with capillaries of the pia mater. And our ependymal cells. And this forms the chori plexus. The chori plexus is found in the floor of the fourth ventricle, the roof of the third ventricle, and the openings of the fourth ventricle. Okay. Everybody with me on that? Yes.
Is the first one supposed to be the lateral ventricle?
I'm sorry, thank you. Thank you. Now, we've already covered this, but the CSF now is going to flow through, I'm going to do this in a vertical before I run out. Going to go from lateral ventricle, ventricles, through the interventricular foramen, or foramina, to the third ventricle. What comes after third ventricle?
The cerebral aqueduct.
Cerebral aqueduct, good. I don't do trick questions, but that was close to one. Cerebral aqueduct to the fourth ventricle. Okay? So now we can exit the fourth ventricle either through the lateral apertures or through the single median aperture, or go down through the central canal. And enter into the subarachnoid space. It circulates around the last picture that I had up on the board, and then it will cross through the arachnoid villi and enter into the dural venous sinuses. Kind of like a concept map with words. And the locations it goes through. Now I know I have it on the board, but just to summarize the sinuses then, the sequence of venous sinuses, flow components, will be superior, sagittal sinus, the transverse sinus, the fluids would be right there, to the sigmoid sinus, to the internal jugular vein. The inferior sagittal sinus will flow into the straight sinus, and then that will go to the transverse sinus. Okay. Easier to see by looking at the diagram itself, but if you wanted a worded sequence, what's in red there will be a worded sequence. All right, so we're done. This is the material that will be on the lab quiz on Thursday. So labs 21 and 22, except for the components of the cross section of the spinal cord. Okay. So no anterior gray horn or lateral white column or anything like that. Just the conus ventularis and the things I drew on the board for spinal cord. Okay, so take a break. I'll pass back your lecture exams and lab exam. And again, you get those 2 extra